Okay, dear guys, let's continue our set of lectures about the cell as a unit of health and disease. So now we will delve much deeper, and some parts will be happened. So it might be very complicated, but still you have to answer to this in tests I'm gonna give you. MCQs. Your practical classes will be held in MCQ way. I'll give you MCQ link. You can answer to this, and my humble request to you, please answer to this by yourself, because previous semester shows me that so when I give common tests, you start to try to help each other, and it decreases your mark because I have no time to over this subject. And please learn it by yourself. And especially, so you're going to be a doctor by yourself. Yeah, you're not going to be a group doctor, and you must learn it by yourself. I understand that most of you will disagree with me. Yeah, that it's not very important, but I know what I'm talking about. Yeah, so that one thing I used to say to my students. So sorry, let me show my face. Where is it? Where is it? Yeah, yeah. I want to say to you that a future doctor is a doctor who knows very deep information. If you know it superficially, you're going to be that doctor. And and previously, I mean, 20 years ago or maybe 50 years ago, doctor is a source of information. Is a source of information, and only your own beliefs or your own knowledge, so is necessary for you. But now, every patient is able to enter the Google. Is able to Google their symptoms. Yeah, you know this funny clip. Yeah, when they said never Google your symptoms. Yeah, but really, so most of patients can be treated by Google. And to be a good doctor, you have to handle disorders better than Google. And very soon will be the situation when how to say it right, when robots Google will replace you. And only one way to not be replaced by Google, it's to be better than Google. You need to understand it much deeper than artificial intelligence. And so I'm sure that very soon, in a few years, might be 10, maybe 20 years, when you will become a good doctor, any doctor, yes, so maybe not very good doctor, but you will be replaced by artificial intelligence. So, and to win this competition with the computer, you have to know it very deep, and that's what we should. That is the purpose of our education right now, to make you better than computer. Okay, let's continue. So in previous lecture we started the explanation of the cell and its relation of cell activity and relation to the pathology, and we will continue it in this lecture, in 3 more lectures. So let's move. Yeah, plasma membrane. So my face bothering me. Okay. Plasma membrane protection and nutrient acquisition. So plasma membrane is composed of phospholipids and proteins and and also it's composed of so carbohydrates, of course, carbohydrates. And we used to think that we have lipids, phospholipids, and proteins floated inside. But actually, our plasma membrane is a little more complex. For example, we have different lipid composition in outside and inside the membrane. For example, phosphatidylinositol it's a part. It's a lipid which opens in an inner membrane leaflet. So because it is important in install phosphate pathway of regulation, so it takes part in our hormone regulation. So okay, we'll discuss it later, but you have to understand that plasma membrane is different outside and inside, even in lipids, even in lipid composition. Okay, phosphatidylindole, one of the most important. Then phosphatidylserine. They are distributed in the inner phase, where it confers a negative charge and is involved in electrostatic interaction with proteins. So when it flips to the extracellular phase, which happens in cells undergoing apoptosis, it becomes a eat me signal. So once again, phosphatidylserine is present in inner phase, but when it's relieved outside, so it gives a signal to our immunity cells to kill it, which is called as apoptosis, so self-killing process. Okay, and glycolipids and sphingomyelin are preferentially expressed on the extracellular space. They are known as gangliosides with complex sugar linkage and terminal sialic acids that confer negative charges. They are especially important in sperm to egg interactions, and they are responsible for cell to cell, cell to matrix interactions, and also they are found in nervous system. Okay, let's continue. Certain membrane components associate laterally with each other bilaterally into distinct domains called lipid rafts. Lipid rafts, so in diagram I will show it to you. Okay, they just frame the specific regions of the cell membrane which perform their specific functions, so proteogas. Okay, the plasma membrane is liberally studded with a variety of proteins and glycoproteins involved in their ion and metabolite transport, very important, fluid phase and receptor-mediated uptake of macromolecules, and cell ligand, cell matrix, and cell-cell interactions. This is a function of proteins of cell membrane. I said about it in the beginning, in the first lecture, a little bit, but now we'll stop on it. So, for example, okay, this diagram says, as I promised to you, the specific part of cell membrane, known as lipid rafts, it possesses some specific lipids. It might be different from cell to cell. Absolutely, it doesn't matter. So, we are not going to learn it by heart, yeah, but you have to remember that we have some lipid rafts, which are typical for some specific parts. And we have glycolipids, so when we see this lipid, and adjoined to this glyco part. Glyco means carbohydrate, yeah. And these glycolipids usually present on the outer surface of membrane, because this glycolipids are responsible for cell-to-cell interaction. I mean, it's receptors. Receptors or antigens, so better to say antigens of our cell membrane. Okay, also inside the cell membrane we can see the transmembrane proteins. These transmembrane proteins they bind extracellular part of the cell with intracellular part of the cell. So function of them were discussed in histology previous semester. Okay, most proteins are transmembrane or known as integral proteins, more relatively hydrophobic alpha helical segments. Okay, we understand it's tough. But main understanding that transmembrane integral proteins binds external part of the cell with internal part of the cell. Okay, let's keep it. Okay. Many plasma membrane proteins function together as larger complexes. This may assemble under the control of chaperone molecules in the area or by lateral diffusion in the plasma membrane. So that, okay, some examples will be shown a little later. Okay, and now let's discuss the transport. How transport in transport inside the cell and outside the cell happens. We have passive membrane diffusion. Passive membrane diffusion is when we don't need any protein, any specific features to pass it out. So, for what we can afford it? We can afford it for oxygen and CO2. Yeah, so we don't need some specific protein, some specific mechanism to pass oxygen inside our cells, because oxygen is always necessary for us. And same to CO2, we need to remove it from our cells, and as well as they removed, so we should give them opportunity to live, to get rid of our cell. Yeah, and usually molecules in which are less than 75 daltons in mass. Dalton is okay. Google it, please. Dalton is a type of mass measure. It's depends on molecular mass. So I hope you're very well in chemistry and you understand what is this. Okay. Um, these particles, which are sorry, non-polar particles. Only hydrophobic particles are permeable less than 75 daltons in mass and through the cell membrane. Polar particles are restricted from passing out. So to pass the polar compounds, we need another features. Let's show it later. Okay, but passive membrane diffusion is possible for O2, CO2, and other small molecules less than 75 daltons, which are non-polar. Once again, non-polar. And for example, examples of it is some hormones like steroid hormones, some lipid soluble compounds like halotan. If you ever been in surgery department, you remember that for anesthesia we use halotan or some other. You inspire some drug, yeah, so which permeable in your lungs and then later in your brain, and which seduce you. Okay. Let's continue. But most of for a lot of compounds, we have some specific features, some specific tools, and they are known as carriers and channels. Carriers and channels. And you remember this. You remember we have passive transport and active transport. So channels is a typical example of passive transport. When so channel is so sorry, I have to remind myself. No, it's not present. So channels are proteins which work as a like gates in our cells. Yeah. So when it's closed, so this no compounds can pass through it. But when it's opened, so some compounds can easily pass through it. So and these channels. So one of the most well-known channel is a sodium channel. So when it's opened, sodium easily can pass inside the cell. When it's closed, sodium is restricted. It's very important in nervous system. You remember this potential of the cell. So when we pass the electric signal along the cell through the nervous fibers and through the muscles also. Okay, but carriers are usually related to active transport. So carriers need some ATP to transport compound from inner side of cell to outside, or opposite, from outside to inside. Yeah, that's a feature of carriers and channels. Okay, examples are shown here. Please read it. That's okay. Concentration and or electrical gradient between the inside and outside of the cell drives solute movement via passive transport. So when, for example, even compounds with a charge, but if they're present in higher concentrations, they can easily pass by passive transport. Okay, we'll discuss it in a particular topic. Okay, let's move forward. Um, Receptor-mediated and fluid phase uptake. Uptake of fluids of macromolecules by the cell called endocytosis. You know this endocytosis, different types of endocytosis. You learned it as pinocytosis, phagocytosis, and receptor-mediated endocytosis. Yeah. So here we start with receptor-mediated and fluid phase uptake. So here I want to stop on this diagram. So actually here in previous slides you can find all the description of this, but I'll explain it in easy way. Look, this is our carrier protein. Usually carrier proteins are known as pumps. With the help of energy they just rotate it, and some molecule might be glucose, might be some other, never mind, a lot of different molecules which are passing by this mechanism. So when some specific components, okay, let's explain to an example of glucose. They grabbed by this carrier protein, rotated with help of energy, ATP energy, and moved inside the cell. Okay, channel. Channel is a gate which might be closed, and impermeable for this compound, or may be opened. And then by passive diffusion, along the concentration gradient, it passes inside the cell. You know it, so just I don't stop on it because you already should know it. If not, if you feel that something is absolutely misunderstandable by you, please repeat again your histology notes. Okay, and then we have endocytosis, and we have caveola-mediated endocytosis. Caveola-mediated endocytosis, it's not related to receptors. So when digestion goes with help of caveolin protein, which just surrounds it and forms the vesicle inside the cell. It's endocytotic vesicle, typical endocytotic vesicle, and this endocytotic vesicle might continue migration down. It might form the lysosome later, or whatever. Yeah, so a lot of different features. Okay, we have receptor-mediated transport, also known as clathrin-dependent transport. Clathrin-dependent transport, when we have receptors on the surface, and when they bind with a specific ligand, so it starts to invaginate. It starts to invaginate and forming the coated vesicle. Coated vesicle coated with clathrin. Clathrin is a specific protein which, so same as caveolin, it helps to close it. But it's different mechanism, and that's why we distinguish this caveolin and clathrin dependent transport. And this is necessary to ingest huge molecules like proteins or something else, which cannot be ingested by channels or carrier proteins. So usually it's a protein, and it's ingested inside, and then early endosome and late endosome, as also discussed in histology. So it enters to form the lysosome, and lysosome. Lysosome late endosome fusion vesicle, so you need to know this mechanism. So it fuses and forms a final lysosome, and we can digest this protein. And we have phagocytosis, also phagocytosis. So phagocytosis is when we ingest extremely huge particles, like microbes. Yeah, especially it's necessary for our immunity cells. So when cell ingests the microbe, forms a phagosome, and then phagosome so get fused with lysosome, it forms a phagolysosome. And then so digestion happens here, so bacteria get digested into small particles, and all necessary compounds, all necessary food, yeah, or something, water, saline, doesn't matter, so it gets fused with the cytoplasm, but everything else so remained inside the phagolysosome, It's known as undigested residual material. That's what happened during the phagocytosis. It's it's just a repetition of what you already know. And we have transcytosis. Transcytosis happens with like this. It's caveolum mediated transport. So when vesicles just entered inside the cell and then transport down. It's not really in blood vessels. You know it. You remember it from the endothelia of capillaries. Yeah, you remember that we have different types of capillaries, and in first type of capillaries, yeah, that we have transcytosis as a main feature of this endothelial cells. We can transport the components of blood from blood vessel into the connective tissue under the blood vessel. This is known as transcytosis. Okay, let's move forward. Cytoskeleton. So cytoskeleton is also usually missed part of the when you study. So usually cytoskeleton is just passed out during your study. Okay, components of cytoskeleton. So you understand, cytoskeleton is a part which is like skeleton for the cell, which perform its shape, perform its activity inside, yeah, and it possesses three different filaments inside. They are known as actin filaments, intermediate filaments, and microtubules. Let's talk about them in details. Okay, that's structure. Okay, actin filaments. Actin filaments are fibrils 5 to 9 nanometers in diameter formed from the globular protein actin. It's a globular protein. Globular means water-soluble protein. But later this globular protein became straight and is water-insoluble. It is the most abundant cytosolic protein in the cell. So it's G-actin monomers non-covalently polymerize into long filaments into F-actin. So as I said, G-actin is water soluble, and then they fuse together to form F-actin that intertwine to form double-stranded helices. So they are most commonly present, and they perform the function of activity, and especially in muscles. In muscles, you know that actin work along with myosin, and actually same actin which present in muscles is present in each cell in our body, and they just control cell shape and movement. Intermediate filaments. Intermediate filaments are most variable type of filaments in our body. They usually present in different cells with different features. For example, vimentin, they're present in mesenchymal cells. Desmin, present in muscle cells. Neurofilaments, they're present in axons of neurons. Glial fibrillary acidic protein, they're present in glial cells that support neurons. And cytokeratins, they're present in epithelial cells. So, why is it so important? Why we are talking about these intermediate filaments? Actually, we can use these intermediate filaments as a marker of might be tumor. Yeah. Just imagine you're as a physician. Yeah. So some patient came to you and said, "So you see here, I have some, I don't know, tumor, some tumor present on your skin, and you have to confirm where this tumor grows out." Yeah, sometimes it's very hard to distinct it. Now, for example, in case of metastasis, yes. So, for example, you need to confirm where this tumor came from, and at least you have to understand it's mesenchymal tumor, or might be neural tumor, or glial tumor, or muscular tumor. Yeah, and we use specific stains to mark these specific intermediate filaments, and this is nowadays it's one of the most usable type of staining of tumors to stain these intermediate filaments. Okay, we'll discuss it in a topic about tumors. It will be far away, so next week. Next weeks in the middle of semester we'll discuss it. Okay. Intermediate elements are found in a rope-like polymerized form and primarily serve to impart tensile strength and allow cells to bear mechanical stress. It's pretty obvious actually. All the filaments particularly handle this function. Okay. Microtubules. Microtubules are also known as thick fibers. They're made by polymerized dimers of alpha and beta tubulin. And they are arrayed in constantly elongating or shrinking hollow tubes with a defined polarity. They have this designed positive and negative charge. Yeah, so like amino acids, you know that some part of amino acid is positive, another part is negative part, and this negative charged parts present on one side, and on another side present positively charged part. Okay. The negative charged part end is typically embedded in a microtubule organizing center. So this is like scaffold for the microtubules growth. Microtubule organizing center is you heard about it. It's centriole. It is centriole, and positive end it elongates during the microtubule organizing center, producing new microtubule. So, and one of the best examples, it's when producing all the fibers which connect to the in metaphase, forming the metaphase plate. Yeah, you remember this. So how is it called? You remember? So let me remind me. Yeah, I found it. It's spindle fibers. Yeah, so like spindle fibers are typical microtubules, which starts from the microtubule organizing center, centromere, and connected to centrosome. Sorry, from centrosome and elongates to centromere with the help of kinetochores. Yeah, that's typical microtubule growth goals and positive charged end of microtubules attached to the kinetochores of the centromere. Okay, and another function of these microtubules is the mechanical support for sister chromatid separation during mitosis, as I said right now. Core of primary cilia, single non-motile projections on nucleated cells that help regulate proliferation and differentiation. This is cilia, and the core of motile cilia in, for example, in bronchial epithelium or flagella in sperm. They are also made by microtubules, and this we also discussed in histology. And now another chapter is cell-to-cell interactions. Cell-to-cell interactions are especially typical for epithelial cells. They are when cells are closely joined together. When the closely are joined together, we need to join them with something. We need to tie them, yeah, and we tie them with occluding junctions, anchoring junctions, and communicating junctions. Occluding junctions are known as tie junctions, and anchoring junctions are desmosomes, which also sometimes refers to tie junctions, to be fair, and communicating junctions are known as gap junctions. Occluding junctions. Okay, I'll show it to you in this diagram. Occluding junctions, they're present here. So wait a bit. Yeah. Occluding junction is present here as a tight junction. You see, it's like proteins which sew them together, which connect the apical portion of the cell with the lateral portion of the cell, lateral binding sites. And but under them we have a belt desmosome, which is occluding junction. So which bind them insulatively, so which forms the insulation between cells. And also here we have desmosomes, which bind cells like in in a way of button-like. Yeah, so hemidesmosome in one cell binds hemidesmosome of other cell, and together they form one common desmosome. You see, this desmosome contains what? They contains out of cell connection, which connects one desmosome to another desmosome, and inside the cell. They possess fibers that connects the cell to the cytoskeleton inside the cell. It usually connects to actin filaments, but also is able to intermediate filaments connection and sometimes even microtubules connection. And these desmosomes are connected from one side. It connects the cytoskeleton inside the cell, and from another side it connects to another, the hemidesmosome. Hemidesmosome means half of desmosome, and you should have talked about it in previous semester in epithelial cells. Okay, without stopping on this too much, so hemidesmosomes also present on the basal compartment of the epithelial cell. This basal compartment, it connects, cytoskeleton of the cell with not with another hemidesmosome like in lateral compartment, but with basement membrane. So here you see here you can observe the basement membranes on under this, and integrenes of this hemidesmosome grabs the basement membrane components and allows the resting of epithelial cells on basement membrane. That's a point I want to say to you. Okay, and another very important communication between cells, there are gap junctions. Gap junctions are present here, also in lateral compartment of the cell, and they communicate one cell to another. So cytoplasm, so liquid component of one cell is connected to liquid component of another cell, and ions, water, and water-soluble compounds easily can pass through these gap junctions. It happens to make it like a community of cells. Yeah, so one cell might grab some saline solution. Yeah, another cell might grab some glucose. One cell can survive without saline, another cell can survive without glucose, and they just exchanging this material between each other. And that's the point. But what happened when one cell is stopped communicating with other cell? So, for example, when this interrupted, for example, I'll try to draw it. Yeah. So I want to draw it. Yeah, okay. For example, this cell is dead. What happens? How neighboring cells understand that this cell is dead? With help of these desmosomes and gap junctions, we can feel this cell can feel that here is a gap, and what it leads to. So actually, activization of a lot of here also activization of when this desmosome get untied, so it leads to activation of mechanism inside the cell, and cell tends to reproduce this cell. So one dead cells leads to proliferation, cell division of other cells. That's what is point of the that's point of this communications between cells, cell to cell interactions. So here the information in more scientific way explained. So I explained it in a very broadly way. Yeah. But I hope you will read it. Pod desmosomes, which you've seen individual desmosomes, also cadherins, and bell desmosomes, which are present like collar of the cell. Yeah. And hemidesmosomes, which also they have integrenes like cadherins, they attach to intracellular intermediate filaments, and those they functional link to the cytoskeleton of extracellular matrix, and they form the focal adhesion complexes. Okay, let's move forward. Gap junctions we discussed. Biosynthesis machinery, that's another chapter of this lecture. So we have a little time, so I'll try to explain it in an easy way. Biosynthesis machinery, is related to nucleus. Yeah, you remember that we have mRNA synthesis and we have primary mRNA and secondary mRNA. And secondary mRNA with its collection of exons of the cell, exons of the gene, they are translated on the rough endoplasmic reticulum. Rough endoplasmic reticulum with ribosomes attached to this. And on this rough endoplasmic reticulum produce proteins. But we used to talk about it in histology and biology in school, yeah. But we need to talk about it. Rough endoplasmic reticulum. So where should I take my place? Okay. They translate mRNA, rough endoplasmic reticulum, and usually produce proteins for extraction. To extract it somewhere might be to produce proteins for outer surface of the cell, and also might produce proteins to function inside the cell. So, okay, but what if proteins are produced, which are produced by rough endoplasmic reticulum, became wrong? What happened? Actually, these proteins became digested by proteasomes. You remember in previous lecture I said about the proteasomes, which are enzyme complexes which destroy the proteins into amino acids and small structures. So, okay, it's more explained later. So these misfolded proteins. And if amount of misfolded proteins is too much, if too much proteins which are produced by cell are misfolded, so it leads to endoplasmic reticulum stress. It's this condition leads to our next chapter topic known as cell death, apoptosis. Apoptosis is a programmed cell death. Okay, don't worry, we'll discuss it in details. So sometimes endoplasmic reticulum editing function leads to disease known as cystic fibrosis. Cystic fibrosis is it happens due to mutation in CFTR gene. It results in the loss of single amino acid residue, phenylalanine. Only one single residue changes, leads to this disorder. And it leads to dysfunction in chloride transport and leads to increasing of viscosity of bronchial secretion and also pancreas secretion, and leads to airway infection and pancreatitis. So recurrent pancreatitis. Okay, we'll discuss it in genetic disorders pathology. So in a few lectures after this. Golgi apparatus. You already know what is Golgi apparatus function. It's very closely functioning with the endoplasmic reticulum. So vesicles from endoplasmic reticulum are set to Golgi complex, Golgi network for modification of protein, and also to mixing up of proteins with lipids or carbohydrates. So that's the function of Golgi complex. Okay, and Golgi complex also in functioning to produce mucus. Yeah, to produce a lot of mucus, which produced by, for example, in our salivary glands and in most of our glands actually. Okay. Smooth endoplasmic reticulum is is a site of synthesis of lipids. Forming so even cell membrane lipids. So actually, Golgi complex is made up of components which are produced by smooth endoplasmic reticulum. And another important function of smooth endoplasmic reticulum is a possession of P450 system. 450 system. A P450 system is a cytochrome function, cytochrome system which able to transfer the water insoluble compounds into water soluble compounds. Especially it's necessary for detoxification. Okay, one of examples. So which example to give? Okay, in time of poisoning, most of our poisons are water insoluble. They're non-polar. Non-polar equals now water insoluble. Yeah, you know. And in this cytochrome P450 system, so handles it and change the shape of the molecule to make it water soluble, to make it possible to release it by kidneys. So SER with this cytochrome P450 system is very abundantly present in our liver, and that's why you might heard about it. Is that a liver is a site of detoxification. We can detoxify our compounds by the liver. So please keep it in your mind. In the further pathologic review, I'll refer to this slide to you later. Waste disposal, lysosomes and proteasomes, and I already explained in basic level. So that lysosomes, membrane-bound organelles, they contain roughly 40 different hydrolases. Hydrolases means they hydrolysis, yeah. So from chemistry, remember hydrolysis. It's decomposition with help of water, and hydrolases are enzymes that function to destroy molecules which are able to be destroyed by water. So they just increase the rate of reaction, and to destroy these molecules. Okay. Okay, that's enough. You know the function of lysosomes. I don't need to talk here. And okay, proteasomes. Proteasomes, which are so proteasomes are targeted to destroy proteins which they are activized by ubiquitin. Ubiquitin protein is a protein of RER. When I said about the RER stress, yeah, I said that proteins which are misfolded, they activize the ubiquitin system, which activize the polymeric proteasome complex, and this polymeric proteasome complex destroys this protein which failed in production. And they digest proteins into small 6 to 12 amino acid fragments that can subsequently be further degraded to their constituent amino acids and recycled or presented to immune cells in the context of major histocompatibility complex. Okay, I understand it's too tough. But main point is that a polymeric proteasome complex destroy proteins which are failed to be used in our body. And they are present not only to destroy our own wrong proteins, synthesized proteins, but also when we ingest some viruses or bacteria, and to present these parts of these bacteria and viruses to the surface of cell to be recognized. And that's how our immunity works. And we'll discuss it in topic of immunity disorders and also in inflammation a little bit. Okay, here it shows phagocytosis. Yeah, so what happens during the phagocytosis? So digestion of bacteria goes, destruction of this bacteria. So it fuses with lysosomes after this, and residues of this bacteria so might be extracted outside. That's a function of lysosome, actually, to destroy everything which were digested in our body, in our cell. Sometimes the lysosomes actually can destroy our own parts of the cell, and with the LC3 mechanism we can frame the part of cell which should be destroyed, and lysosome fuse with this part with mitochondria you see here, so might be some other organelles which need to be renewed. And then happens same process, so we treat our own organelles like some alien cell, and we destroy it and exocytose it, so remove it from our cell. That's a function of lysosome. But what about proteasomes? So proteins which are produced in rough endoplasmic reticulum, they get controlled by chaperones. They are specific proteins which assist in folding of proteins. But if this folded protein get unfolded, so might be by age, might be by ultraviolet, might be by heat, reactive oxygen species, and might be it's initially produced wrong, yeah, so by genes. So it's, and this denaturated protein get destroyed. So with the help of E1, E2, E3, this free ubiquitin. So as I said, free ubiquitin molecules, they form multiple ubiquitins, and they activate the proteasome. And this proteasome just start to destroy this protein. But if these proteins, misfolded proteins, are too much in number, it leads to formation of ER stress, endoplasmic reticulum stress. And too much of these proteins, when they produce, they start the process known as apoptosis. In much amounts, much deeper, we'll discuss it in next lectures. I mean, so after 2 lectures, we'll start to talk about this. But keep in mind that misfolded protein is a cause of cell death. Okay, that's it in this lecture. So thank you for attention. I hope it was interesting. So see you next time.